Thank you.
We have to move on to the next lecture by Professor Jacques Descotes from the Department of Pharmacovigilance and Poison Center, Lyon University Hospital from Lyon, France. Jacques, thank you for coming.
So I've been asked to very briefly overview the immunotoxicity potential of nanomedicines and what are the clinical aspects. It will be easy because we could say we know nothing, or nearly so, on clinical aspects. And regulatory aspects are the future, obviously. I don't know where I have to push. Okay. Well, you all know that nanomedicines are widely heterogeneous molecular entities, and obviously, as it has been already said by the first speaker today, we know that a number of characteristics, physicochemical characteristics, are likely to impact on the effects of nanomedicines or nanoparticles on the various components of the immune system. What is also important is that nanomedicines are potentially useful to diagnose or treat a wide variety of pathological conditions, which means that the immunotoxicity, the prediction of or the assessment of the immunological safety of nanomedicine should have to take into account the various conditions or modalities of use of nanomedicines. And finally, the information we have shows that a number of nanoparticles have the ability to trigger effects on the various components of the immune system, like triggering an inflammatory response, interact with immune cells, activate the complement cascade, or facilitate antigen-specific hypersensitivity and so on and so forth, obviously. And so there is a large body of immune effects that have been associated with nanoparticles. Well, for an immunotoxicologist, it is important to say that it's not because a drug or a compound, a chemical, induce changes, immunological changes, that it is immunotoxic. Immunotoxic means that you induce changes in the immune system that are likely to induce some adverse effects in treated humans, which is, very, very different, obviously. The immunotoxic potential of nanomedicines presumably can be subdivided into 4 categories, and it is currently subdivided regarding other medical products. And that is, these categories are immunosuppression, immunostimulation, hypersensitivity, and autoimmunity. Immunosuppression, it is well known with the long clinical experience gained with immunosuppressive drugs that you can anticipate more frequent, often severe, sometimes atypical infections. They can be caused by bacteria, viruses, fungi, parasites, and also the other major consequence are most frequent virus-associated neoplasias. It is often said that lymphomas are the typical virus-associated neoplasias. In fact, cancer of the skin and lips are far more frequent than lymphomas that are mostly B-cell lymphomas. And there are a few data showing some examples of immunosuppressive nanoparticles or nanomedicines, and I just gave you some examples, and I'm sure you know many other examples. Many I'm not so sure, but at least several other examples. And obviously, we can consider that nanomedicines may prove to be immunosuppressive because of targeted or nano-delivery engineering to produce intended immunosuppressive agents or immunosuppressive pharmaceutical formulations. because, and this is very important for immunotoxicologists, and based on regulations I will come back to in a few moments, the sequestration of variable duration with the immune cells, lymphoid organs, or tissue that can potentially result in immunosuppressive effect via various mechanisms, including inhibition of phagocytosis by macrophages or impairment of other immune cells. And as always, it is impossible to be sure a new compound will not have unanticipated immunosuppressive effects. So that's also a possibility, I assume, with any new nanomedicines candidate nanomedicine, as it is absolutely true for any new drug candidate. Immunostimulation or immunoactivation, the anticipated adverse clinical effects are primarily acute systemic complications due to cytokine release. It could be mere flu-like reactions, and the very good examples are vaccine boosting, when you have boosting injections. In children, for example, they have fever, they feel not very well. This is a flu-like reaction. But these reactions can be far more severe and be really acute cytokine syndromes, but these reactions will be largely due to IV routes or sub-Q to some extent. Immunostimulation can lead to more frequent immune diseases, not always, but it's a possibility, absolutely well documented. More frequent hypersensitivity reactions to environmental allergens. This is something that has been known for many years, that has been largely overlooked, and it's quite funny to see that, in fact, it regains some concern due to some nanoparticles. And finally, immunostimulation can induce inhibition of cytochrome P450 pathways via IL-6 release. Then this is a potential cause of drug interactions. And obviously, there are examples of nanomedicines that can be immunostimulatory or immunoactivating. And as it is often the case with immunology, immunopharmacology, or immunotoxicology, you can have examples and the opposite examples, so that it is quite difficult to clearly elucidate what is really the context or the concern with the new product. And an example, I told you about this increased risk of allergic reactions to environmental allergen. It has been shown that you can induce, you can enhance sensitization to availability in mice or rats, for example, due to availability due to inhalation of ultrafine diesel, or as well thiolated ketosine nanoparticles. And in fact, exactly the opposite situation has been shown with the inhalation of fullerenes that has been shown to decrease sensitization to ovalbumin. So it's just a question mark, obviously. Hypersensitivity. Well, it is. I heard several comments I was not fully, I didn't fully agree with this afternoon. What is absolutely sure is that hypersensitivity can be either immune-mediated, and these are immunoallergic reactions involving immunological memory and immunological mechanisms, and non-immune-mediated reactions that are pseudo-allergic reactions involving some mediators that can be involved in immunological reactions but are released outside any immune-mediated mechanism. And roughly speaking, we do not have sound epidemiological data on hypersensitivity reactions induced by drugs, but there is a rough consensus suggesting that roughly these two types of hypersensitivity reactions are one half. That means that true immunoallergic reactions are presumably not more frequent than pseudo-allergic reactions. So if we come back to immune-mediated immunoallergic reactions, you can have anaphylaxis, and anaphylaxis is only caused by immune-mediated mechanism and never by complement. Never. Or complement activation alone, direct complement activation alone. Immunoallergic cytopenias are other examples. T-cell mediated organ-specific reactions like hepatitis, pneumonitis, nephritis, toxidermias are good examples of immune-mediated hypersensitivity reactions that are rare but unfortunately potentially lethal. And to the best of my knowledge, I never heard and never read any case report of immune-mediated hypersensitivity reactions due to the nanomoiety of a drug in humans so far. And the other situation is non-immune-mediated pseudo-allergic reactions that could be so, well, it has been asked, obviously, complement activation, direct complement activation is one mechanism, but it is by far not the only mechanism. You can have direct cytokine release, you can have direct histamine release, and therefore these are pseudoallergic. You can have also kinin interactions with kinin degradation, prostaglandin pathways, and so on and so forth. And the major characteristic is that you can never have an immune-mediated reactions during the first contact with the offending drug, but with non-mediated immune reactions, because it is a pharmacological or toxicological mechanism, then it could be a first dose event. And what is important is that clinically speaking, pseudoallergic reactions are not exactly similar to anaphylaxis, let's say, complement activation reactions due to complement activation and anaphylaxis may mimic somewhat, but they are somewhat different. It is absolutely essential to very carefully make a sound medical diagnosis to make the difference, because otherwise we don't know what we're talking about. Well, so regarding immune-mediated hypersensitivity, what can be the concern regarding nanomedicines? What I could say is that, well, just briefly, the immunogenicity of nanomolecules globally could be summarized that way. These are foreign molecules, that is, non-self molecules, therefore they could be immunogenic. But they are really rather low molecular weight molecules. They are non-peptidic chemical structure in most extensive examples. And finally, there is very limited, if no formation at all, of reactive metabolites. Therefore, globally speaking, and being very cautious, I would anticipate a fairly low immunogenicity potential of nanomolecules in general, obviously, and not more. However, it is also important to keep in mind that immunogenicity can result from inadvertently absorbed or purposely bound chemicals or drug moiety, the so-called targeted engineering, and therefore I believe, because the first sentence was rather assumptive and the second has no firm answer, that the immunogenicity potential of any new nanomedicines should be always considered. Regarding non-immune hypersensitivity, nanomedicines can induce histamine direct, that is to say non-IgE mediated histamine release. They can activate the complement and this has been shown with some liposomes and not all liposomes, and that's been shown essentially with pharmaceutical compounds using chromophore as the solvents. And it is absolutely important to keep in mind that these mechanisms, histamine release or complement activations, are really limited, restricted to the IV routes and to drugs that are injected rapidly and high dose. We have a lot of examples showing that even though you have a drug that is known to induce histamine release, vancomycin is a very good example, if you slow down the speed of injections, you decrease the dose, you have no histamine release at all. So, I mean, it is much better to have complement activation due to a nanomedicine than anaphylaxis, true anaphylaxis. Because if it is true anaphylaxis, your drug is dead. If it is complement activation, you have some opportunities to modulate the modalities of administration. Autoimmunity. So, drug-induced autoimmune reactions can manifest as systemic or organ-specific disease. There are very assumptive, to remain polite, data available on potential link between autoimmunity and exposure to ultrafine particles. And autoimmunity is a very complex issue. From a regulatory perspective, it's quite easy because we simply have no models able to predict the risk of autoimmunity due to any compound at all. What could we do about the evaluation? Or what could we say about the evaluation of the immunotoxic potential of nanomedicines? The regulatory setting is relatively easy because there is no guideline whatsoever devoted to the immunological safety of nanomedicine. And there is absolutely no guideline devoted to the clinical immunological safety evaluation of medicines in general all over the world. So my recommendation would be to follow a case-by-case preclinical immunotoxicity evaluation strategy adapted from ICH-S8, which is the guidelines for the immunotoxicity assessment of most, well, I would say standard drugs. ICH-S6R1, which is the preclinical safety assessment of biologics. And I could say that I would not say, I heard early this afternoon that nanomedicines are progressing so fast that it's impossible to keep up. But that's not a new situation. You know, biologics, 20 years ago, biologics are monoclonal antibodies, therapeutic proteins, and so on. And it was also an area progressing very rapidly, and it was very possible to reach some consensus on how it could be, their safety could be predicted and managed. And there are guidelines now. And the ICH-S6 was published in 1995. And we also have an ISO guidelines on the immunotoxicity of medical devices. And it's also true that nanomedicines sometimes, or nanoparticles, are not so far away from medical devices. So we could also get some hints or good ideas from the nanomedicine world. The medical devices world. And one problem is that these guidelines are mostly focused on immunosuppression potential. So we come back to the distinct categories, and immunosuppression. The strategy I propose is, first of all, to conduct a short-term, that is roughly 28 days, repeat dose toxicity study. Obviously, the relevance of the species should be carefully addressed. It is often the rats. The initial assessments will be based on clinical signs during during standard toxicity studies. hematology, clinical chemistry, and histology of many fluid organs. It is the so-called weight of evidence approach in the ICH-S8 guidelines for any drugs. So I understand that nanomedicines are intended to be drugs, so at least for the time being, we could consider that ICH-S8 could apply it, even though some case-by-case deviations could be accepted. I really believe that at least one immune function test, and the prototype test is the T-dependent antibody response assay, the TDAR assay. I think it is absolutely essential because we know so little about the immunotoxicity potential of nanomedicines and how to predict it and to make some assessment that I think it is absolutely necessary. And obviously, it is absolutely necessary to have a good quality dossier of the nanomedicine before starting any immunotox study. There are some shortcomings and pending issues, obviously. Additional immune functions, in vitro, ex vivo, or in vivo, should be necessary, should be assayed, obviously, based on known or suspected immunological effects of the tested nanomedicine. It is important to keep in mind that so far no in vitro assay as yet has been shown to be a reliable predictor of immunosuppressive potential, so there are many uncertainties. One major problem regarding nanomedicines, and I think it's not restricted to immunotoxicity, is that current available assays are not fully validated or standardized for nanomedicines, and this is absolutely essential that this is urgently summarized. With immunostimulation, absolutely no guidelines can be used. The general concept is to say that we could presumably try to use the same type of short-term repeat dose toxicity study for immunostimulation or immunostimulation. Obviously, there are some ex vivo or in vitro assays focused on potential adverse effects that could be used, cytokine release assays, using human cell lines or blood and nothing else, complement activation, lymphocyte proliferation, and also safety immunopharmacology studies because, for example, if you have an immunostimulatory nanomedicine, the question may arise, or should arise, is there a risk for more frequent hypersensitivity reactions due to environmental allergens? With hypersensitivity, no dedicated guideline. My belief, or I suspect, that the preclinical assessment of hypersensitivity to nanomedicines may be less tricky than with small molecular weight medicines due to the lack of metabolic activation, though there is no involvement of reactive metabolites, which is a nightmare regarding small molecular weight molecules. And also less tricky than with therapeutic proteins because current nanomedicines are not humanized, and therefore, as biologics are, and therefore animal models, at least theoretically, should be applicable. And there are a wealth of models of assay. I will not screen them all over, but there are really doors open to test for the hypersensitivity potential of nanomedicines in animal models or using various assays. With autoimmunity, it's easy. There's nothing to say. And finally, I think that the currently available immunological data with nanomedicine suggests immunotoxicity as a possible cause for concern with, I don't know if many is appropriate, with nanomedicines at least. And routine preclinical immunotoxicity evaluation is therefore recommended. Most currently available models and assays used in for immunotoxicity evaluation are presumably applicable to some extent at least, even though standardization and adaptation to nanomedicines evaluation is urgently needed. And finally, obviously, the specificities and modalities of the preclinical immunotoxicity evaluation of nanomedicines is hard to be defined and validated for regulatory purpose, and we hope that someday we will have some regulations regarding the immunotoxicity potential of nanomedicines as we have for biologics, for example. Thank you.
Thank you, Jacques. Questions, please? Yes.
Thank you very much for this presentation. I would like to add even something else. I like it very much, the distinction you make in the hypersensitivity between the immunological and non-immunological, the memory versus the kind of innate responses. And I think that we should consider also, as you point, the case by case, because it's not the same a nanoparticle core that basically, if it induces something, will be only an innate response, mostly by phagocytosis and not with the lymphocytes, and then one that is a final product. And depending on what is the product, then maybe we could drive the attention to one part or the other. So it's not the same if you have an antibody, for instance, coated to make a targeting, or if you have an immunosuppressor drug or a cytotoxic drug that can be released or not and have so the adverse effects that they are of the product that accompany. So I think it's very important to make this distinction also of what is the core and what is the additional molecules that they are involved in the final nanomedicine.
Yeah, I fully agree with you. I think that presumably this will be a nightmare for regulators to try to reconcile all the differences, the characteristics of nanomedicines to make useful guidelines and not a very superficial one or very obsessional, detailed one. So I think it will be absolutely difficult because obviously this is primarily case by case. I absolutely agree.
Jacques, you said that immune and non-immune anaphylaxis are very different. In other words,
not very different. Not very different. They are, well, when you are well trained, you can make the difference. Just to give you an example, we've been involved in it at different stage of this. I remember once I got a phone call from a company selling radio contrast media, and they had a problem with a product in clinical hold by EMA, and they said we have, I don't remember exactly, 21 or 23 reports of anaphylaxis, and we would like you to review this report and these reports. And I reviewed these reports, and my conclusion was that at least, out of 23, 21 were absolutely, could not be anaphylaxis. And the two others could be, I don't know. So that really makes a difference. And I said, I believe this is pseudoallergy, pseudoanaphylaxis. And I would not be very surprised that it is due to direct complement activation. And Janus showed that it was direct complement activation. So, and I mean, never believe medical articles or medical doctors when they say this is anaphylaxis. Because I could show you many papers published in very high impact factors journals with false diagnosis of anaphylaxis. So that means that there are differences, but they need some expertise, I would say, medical expertise.
Thank you very much, Jacques.
Thank
and
you.
Then we go to the next speaker.